Okay, recording. And now share screen. Okay. And anytime that I'm not, like if you're not seeing what I seem to be talking about, just unmute and let me know and get that fixed. But I am going to go back to there's a couple slides from extracellular matrix and tissues that I want to talk about a little bit. And then the rest of the time, we will be on the cancer slide set today. I'm going to turn down my sound just a little bit. Okay, so this particular slide here and this one are the ones that I want to talk about before we go on to cancer because they're kind of related. So we had talked about last time the concept of having stem cells that are not differentiated and they have potential to then turn into any other kind of cell. And that ability has a particular word or a particular vocabulary word that I want you to know called pluripotent. Pluripotent. So they're like multi-powered. And if we say a cell is pluripotent, it has the potential to give rise to differentiated cells. So remember that these cells over here that are doing specific jobs, those are differentiated. And these can actually do specific tasks for the body, like store fat or send signals or eat dangers or contract or provide support to neurons. These would be differentiated. These can't do anything, but they have all of the potential. And remember that what part of what makes cells go through this transition from having all the potential but no function to having a great function but locked into place is that they basically are going to inactivate certain genes. If you looked in the genome of any of these cells, they would all have the same genes, but the inactivation of certain sets is essential to take a stem cell and get it to differentiate into a cell with a high functionality, but less potential. So the more that scientists have learned about this process of how do stem cells develop and differentiate into the functional cells, that has provided a lot of knowledge about how can you reverse the developmental process. And that's what this slide is about. This slide is about reversing differentiation. Reversing the differentiation process to kind of move backward toward a stem cell. Just call it backward. Um, and it used to be, so when I was your all's age, the only way to do that was to start with embryonic stem cells, um, basically from fetal tissue or embryonic human tissue. That was the only way that scientists knew how to go backward then. Um, with better study of what are the types of genes that you would need to turn back on in a cell, scientists now are able to take differentiated cells or partially differentiated cells like a fibroblast. So a fibroblast normally is going to make fibers that help build connective tissue, and fibroblasts are found in skin, so that's a good source of getting a partly differentiated cell and figuring out what are the genes to turn back on in this cell and induce it, or force that fibroblast to become a pluripotent stem cell, to kind of go backward in its development. And then once um, scientists are able to make an induced pluripotent stem cell, there is then the potential to turn certain other genes on. So find the particular gene sets that are needed for turning this induced pluripotent stem cell back into these specific tissue types. So by using this process, we can move toward rebuilding new tissues um, for people who may need them in a therapeutic sense. There aren't that many people that are suffering from lack of fat cells and needing those in a therapeutic sense, but being able to regrow neurons or being able to regrow muscle is really a big deal and would help a lot of people. So this is not I wish I could spend more time on this overall topic, but the words pluripotent and what that means, the major functional differences between true stem cells and differentiated cells, and the ability to turn a partly differentiated cell into an induced pluripotent stem cell and then have the chance to regrow functional tissues. That's a huge um, breakthrough field currently in medicine, and you're likely to, as you study more in biology, and especially if you're going into human medicine, um, this kind of research and this kind of process is going to, in your all's lifetime, really change how we can treat people with degenerative diseases, because scientists are learning so much more every year about how to regrow these tissues. And it largely has to do with gene regulation. How do we turn back on genes that have been silenced and give potential to build a new stem cell, and then um, do some particular re-silencing of genes to turn them back into these particular tissues. So I just wanted to get you all that little bit of information about stem cells generally before we go on and talk about cancer next. I need to save this project. Okay, and then go to cancer biology project. This section, again, we could have a whole class about cancer. I think that would actually be one of my and Dr. Vaughn's dream upper level classes someday. But in cell biology, we get this one lecture to talk about cancer. So what I want to do with the time that we have to talk about cancer is to talk about some themes of why cancer develops, and then look at a few examples of molecular details that could then apply to a lot of different cancer uh, mechanisms. So there's some slides that we're going to go through quickly and some slides that we will spend a lot of time on. So in the beginning here, um, I know that you all have a general understanding of what cancer is. It's cells growing uncontrolled in a way that they're going to harm other tissues. And it actually takes several different things to go wrong um, for cells to develop a cancer state. So I want to use this slide to kind of make a list of what are the major events that have to happen for an organism to develop cancer. So the first major thing that has to happen is there has to be a dysregulation of cell cycle. Cancer is always going to come back to cells dividing too often, or dividing in the wrong direction, or losing their ability to monitor their DNA for mutations. So there's always going to be some kind of dysregulation of the cell cycle. And what you learn in this class gives you the tools to understand generally how that happens in all different kind of cancers. So something goes wrong with the cell cycle. And then cells are going to divide inappropriately. Inappropriately. And that's probably the most obvious part of cancer. It's a cell division problem. So cells are going to divide inappropriately. And that's kind of the key to what cancer is. But for cancer to actually become a danger to an organism, those cells have to become a problem. In some way, the cells have to become a problem for other tissues. So either they are going to, this is inelegant how I'm going to phrase this, but they could push on or invade other organs. And sometimes that's the problem, that a tumor is encroaching into another organ and taking up its space. Sometimes the problem is that the cancer cells are using up resources that are needed by other cells. And those are the two major problems really from cancer. Both of those problems are requiring, in general, that the cancer is going to in some way move out of its original location. A little bit more room here. Both of those are going to require that the cancer, put this in blue, that the cancerous cells both multiply and in some way invade others' space. And we can see here an example where there is a cancer in an epithelial tissue. We've got all these cells joined together into some amount of layers, and the cancer has spread and multiplied downward, and it has encroached out of its original location, and then we have cells kind of breaking off and migrating other places in the body. The bigger a tumor gets, and I know this is probably obvious, but the bigger a tumor gets, the more likely it is to push into or invade other organs or tissues. And then this part of breaking off and then migrating to new locations, that can seed other tumor, SEED, seed, start other tumors in the body at other locations. And then in general, the more cancer cells that you have all in various locations together, they are going to use up resources used by the other cells need. So the other couple of definitions that I want to get in here have to do with these words benign or malignant. All right, benign in green. So you may have heard of a benign tumor. So a tumor that is benign is still, it's still in one clump together. So if this tumor had not started breaking off cells to go elsewhere in the body, we could say that it was benign. So in a benign tumor, the cells are still together. I feel so unarticulate this morning. The cells are still together um, in one spot, and that tumor, if it's benign, it hasn't done damage to other tissues yet. And if you have to have cancer, benign is what you're hoping for. So you hope that all the cells are together in a clump. They have not spread elsewhere in the body, and the clump so far has not caused a lot of damage to the rest of the body. Um, Detecting tumors that are still at the benign state is difficult if the lump, the clump, the tumor cannot be felt from the outside. Most people that have benign tumors, it's going to be discovered because it can be felt or observed from the outside of the body and discovered before the point that cancer cells break off and go elsewhere. Hans, if you ever get to meet Hans' mom, she has a scar in her neck because she had a benign tumor on the front part of her neck, and it was discovered because it's, made a bulge from the outside. And when they did the surgery, it was still together in one solid mass, and they took it out, and she didn't really have that much problem from it. But what you don't want, if you have to have cancer, what you don't want is to have malignant cancer, and that's referring to cells have migrated. Cancer cells have migrated from the original tumor, and that's a much more dangerous situation. Much more dangerous situation. These definitions also have to do with knowing what treatment options people may have. So if a tumor is benign, it could be removed just through surgery. If a tumor is, and possibly, depending on what type of tumor it was, you could do surgery and you could do radiation because just the one spot where the tumor was. If a tumor is malignant, um, you still would want to take the main mass out of the patient if you can, if that's not going to harm the surrounding tissues. So you would still probably have surgery. Um, but if the cells are spreading elsewhere in the body and you don't even know potentially where they went, that's when patients would need to have chemotherapy. So the difference between benign and malignant also relates to what kind of treatments would people have for their particular cancer. And probably some of you already know all of that. And if you did, I appreciate your patience, but in case those kind of words have been floating around in your mind without a specific definition, There's the specific definitions. So when cells start to have a cell cycle problem, use this picture to describe this. When cells start to have a cell cycle problem, it's going to come from a mutation. Um, and the mutation that gives a cell cycle problem, if it's going to lead to more cell division for that cell, by definition, that cell that has that particular type of mutation in its cell cycle genes is going to have an advantage over the neighbors. And so that cell is going to divide more often, and it will have more offspring that have that same cell cycle mutation. And because these are dividing more often, they are likely to get additional mutations over their neighboring normal cells. So first of all, it takes multiple mutations to really lead to cancer. It's not just you have this one mutation and it's going to cause cell cycle problems resulting in cancer. It takes multiple mutations are required for cancer development. So if someone starts off with a certain mutation that they've got, we call that they have a risk factor for cancer, and that might mean that they're kind of starting out at a disadvantage because they have a pre-existing mutation that makes cancer more likely. But it takes multiple mutations to get cancer to a level where we really have a lot of proliferation, and then we have invasion. So that encroachment on neighboring tissues, that's called invasion. We should go back and put that over here on this side. We'll have invade other space. Invasion is this concept there. So the other thing that's really helpful to understand about cancer is this fact, which I'll write over here, which is the more often a cell divides, the more likely it is to have new mutations. The more often a cell divides, the more likely it is to get new mutations. And that's because every time a cell divides, there is a chance for DNA copying errors. Every DNA polymerase has a natural error rate that's just part of its existence. And the more that DNA is copied, the more chances there are for mutations. So that leads to two other facts. Because epithelial cells have to divide often to provide new epithelial linings, they are already set up on a program to be dividing often. And every time that an epithelial cell or any other cell goes through a cell division cycle, there's a chance of DNA copying error. So epithelial cells themselves are the most common source of cancers. I think that's worth writing on here. Epithelial tissues are one of the most common sources of cancer. And then the other kinds of cells that have to divide very often all through your life are also blood stem cells. You have to always be making new blood cells, white blood cells, red blood cells. They have to divide very often, and that's why leukemias, cancers of the blood cells, are also pretty common. So if you look at what kinds of cancer, what kinds of tissues tend to start cancers in people, it's a lot of epithelial tissues. So breast cancer comes out of breast duct linings. Skin cancer comes out of epithelial cell divisions in the skin. Colon cancer comes out of the lining of the intestine. It's an epithelial tissue. And leukemias come from blood stem cells dividing a lot. And those are really common types of cancer. So when cells are dividing a lot, they're more likely to get a mutation, and that tends to be in epithelial tissues. And when a cell gets a mutation that causes it to divide a little bit more often, it's going to have an advantage over the neighbors, and the dividing more often makes it more likely that it will get additional mutations. So this proceeding from, one slight mutation giving a slight division and survival advantage tends to cause a snowball effect. So it does take multiple mutations, but having one mutation makes others more likely to get from a cell that's dividing a little bit too often to the point where we have an invasive tumor. This tumor would be invasive, but it is not yet metastasized. Gosh, that's another word we need to put. I forgot to put, so we'll write that here. I bet you already know what this means. Metastasis. So this picture, this tumor is not yet showing metastasis. It may be malignant and pushing on other tissues and harming them that way, but it's not doing metastasis yet, because what that is when the cells of a tumor migrate to new locations. That's metastasis. So this tumor is still together in one clump. It is invading other tissues, but it has not yet undergone metastasis. So looking back at this picture, this is metastasis, where the cells have left the original mass and they're migrating to new tissues. I need to change what I wrote here about malignant is actually metastasis. Malignant just means that the tumor is causing harm either by pushing on other organs and or using up resources. Metastasis is migrating to new locations. So I'm going to go back and erase this, clarify that. Put in here metastasis. Let's go back and write malignant. Malignant means that there is damage being caused. Metastasis means that cells have migrated. If cells have gotten to the level where they are doing metastasis, we would also consider them malignant, because they're going to so many new locations that in itself is a danger. Sorry about that poor word choice by me in the beginning. Okay, so here we have some pictures of cancer. And on this side, we have a dissected, whoops, a dissected and stained liver that's been stained with a green dye. And we can see that within this liver, there are these huge patches of cells that aren't staining in the normal way. And this is individual liver tumors. And this person has quite advanced liver cancer. And then if you looked at that in a microscopic section, this should be the normal appearance of cells in the liver, and cells in the tumor do not have the normal appearance or function. So something else important to note about cancer cells is that cancer cells are generally going to lose their specialized function. They tend to lose their specialized function, and they are not even acting like the appropriate tissue type from which they came. Um, tumor cells are not doing the normal job that they're supposed to do. They're just growing and dividing, and cancer cells tend to be undifferentiated. They are undifferentiated, and in some ways, they're moving back a stem cell type of state. So detecting cancer microscopically depends on looking for cells that don't have the correct shape and appearance, and clinical lab technicians that do this work, like their visual memory of what cells are supposed to look like, is very important in diagnosing cancer. One type of other risk factor for developing cancer is chromosomal instability. Chromosomal instability. And I would define this as large-scale chromosomal rearrangements. So what you're looking at here is, this is a karyotype, a picture of all of the chromosomes within a particular person's cell that is currently undergoing mitosis. And this would be in prophase, kind of in late prophase, maybe middle prophase. And you'll notice here that they've got some chromosomes that are weirdly long and some that are weirdly short. And this picture helps people like us who aren't uh, experts in cytotechnology to see what's really going on. So when you look over here, each one of these chromosomes has been given a false color. And if you counted these up, there are 23 colors for the 23 pairs of chromosomes. And when you see that a color is, there's two or more colors on one set of chromosomes, it's a chromosomal rearrangement. So if you see this yellow color here, a normal person's chromosomes, there would be one set that's yellow, there would be one set that is green like this. So this is showing that this person or this animal's chromosomes have had a lot of breaks and rearrangements. And their gene segments have just kind of been all mixed up and put back together kind of randomly. So having this happen and having that many breaks and reconstruction in chromosomes, what that's going to do is that's going to causes genes um, to kind of lose their control regions. Or let's just say it causes genes to lose their normal transcriptional controls. This would kind of be like if I took your data figure papers and just randomly cut them up and then put random pieces back together, it's not going to make sense. And some of your sentences would lose the beginning part that gave context to what it's going to say. Uh, so having a lot of chromosomal rearrangements, we call that chromosomal instability, and that can lead to cancer. And one of the most common ways that this happens is when people have radiation exposure. So if you get the kind of radiation exposure that causes double-stranded breaks in DNA, your cells will try to fix the DNA and put pieces back together, but it's normally not going to be the right pieces being put back together. So that's kind of some big picture overview stuff about how cancer tends to happen. And what we're going to go through next is some molecular detail that will be on that these concepts will be on your final very frequently. So there are some definitions here and also some examples of two types of genes that are related to cancer development, and they're called proto-oncogenes is one type, and then the other type of genes that are frequently altered in cancer are called tumor suppressor genes. So the difference between tumor suppressor genes and proto-oncogenes and how you tell them apart, that's going to be a big theme on the final. So this particular section is really important to know in detail. So when we think about genes that control cell cycle, I kind of before talked about pushing on the gas. If we're driving a car, pushing on the gas versus pushing on the brakes. So we can make the car go forward by pushing on the gas and go somewhere, head toward a destination. Or we could put on the brakes and stop the car from moving forward. So when I think about proto-oncogenes and tumor suppressor genes, proto-oncogenes are like pushing on the gas in the cell cycle. Proto-oncogenes are genes that encode any kind of protein that normally would help a cell go through a checkpoint. So we could also say that proto-oncogenes normal function is they're going to help a cell push through a cell cycle, or sorry, push through a checkpoint. They're going to promote surviving, or they're going to promote proliferating and dividing, is the normal function of those genes. So if a proto-oncogene has some kind of mutation that makes it work better, write this down at the bottom. If we have some kind of gene that's normal function is to push a cell through the cell cycle and promote cell division. And if that gene has a mutation that causes it to work better, I still listen to the local radio because I'm just getting old. And there's one of my favorite kind of commercials to hear on the radio is from Steve Huff Plumbing. And their motto is better, faster, cleaner. They're going to do a good job on your plumbing. So if any of these genes that normally promote moving through the cell cycle, more division, faster division, if they get a mutation that makes them work better or faster, that kind of mutation in this kind of gene is going to lead to too much survival, too much cell proliferation, and a higher risk of cancer. This would be like your gas pedal getting stuck. So we usually, when we think about the whole world of mutations, we don't usually think that we don't usually think that mutations make things work better, but there are some kinds of mutations that do that. And those are called gain of function. Those are gain of function mutations where something works better, faster, more, and they tend to be dominant. That kind of mutation is a dominant mutation, and here's why. So here's, we have two copies of whatever this proto-oncogene is. So an example could be a cyclin. Use cyclin as an example. That's a cyclin gene, let's assume. And we've got two copies of the cyclin gene in a normal cell. And if one of them gets a dominant or a gain of function mutation, and that one's going to work better, it doesn't matter that this one over here is still normal. Like this one's on speed and it's going to work better, faster, cleaner, I guess, and cause the cell to have too much cell survival or too much proliferation. So this is like if I use an example of employees. Employees, say that you work at a restaurant and you've got two waitresses, and one waitress is pretty good, does their job at the appropriate rate, but one of them's just amazing and like can do serve twice as many tables, um, better, faster, cleaner. It's that waitress or waiter could be activated. And just having one that's overactivated is going to give you significantly more waiter waitress activity. Get the job done better. That would be like a proto-oncogene. So this next slide is going to go over how in the world do you ever get a mutation that makes something work better? Kind of rare, but it does happen. So this is called mechanisms of oncogene activation. We could also say ways to get activating or gain of function mutations. How does that ever happen? It does happen. These are the possible ways it could happen. Make this scoot over. So one way that you could have this happen is that there's a mutation in the gene that codes for this protein, and it literally, that mutation ends up having a better function. The protein is actually functioning better. That can happen. It's pretty rare. Most mutations in a given gene are not going to make it work better, but it's possible. It's possible. So that's one way. A more common way is that you don't make the protein individually better, but you get more copies of the gene. You just get more copies of that gene. So same gene, no mutations, but if you suddenly got 3 copies of it, we call that gene amplification. And each copy is making the normal protein, but there's just a lot more of the normal protein. So if this was a cyclin, and you suddenly have a lot more cyclin, that would be like you've suddenly got 3 waitresses, not one. This case, you would have like a, I don't know, superstar waitress that's just better than the others. But in this case, you just have more by gene amplification. Um, a related way to get the group of proteins to work better is that you get behind a strong promoter. And call this strong. Ah, no. Strong promoter. This gene that gives us the normal protein, it ends up being controlled by a strong promoter. So you get more transcription, or you get, I'm just going to focus on this part. You get more transcription of the gene, and you end up having more protein made. It's not because you've got more copies, but you've got control suddenly by a strong promoter. That's going to happen by chromosome rearrangement. Like this promoter shouldn't be right here; it should be somewhere else in the genome. But by chromosome rearrangement, it ended up in front of this gene for cyclin, and now we're making a lot more cyclin, and that's going to lead to a dominant or a gain of function mutation. Let's put this one in parentheses. No, for this class, knowing these 3 ways is good for this class. This one takes longer to explain. I don't want to spend the time today. So this kind of activation of a gene that normally causes a cell to go through cell cycle in a forward direction is promoting division, it's promoting survival. It only takes 1 copy getting that mutation to cause the effect. So you only have to have 1 copy changed if it's a gain of function mutation in an oncogene. Now we're kind of going to look at the reverse situation, a different class of genes, and these are called tumor suppressor genes. So if proto-oncogenes' normal function is to promote cell division, tumor suppressor genes' usual function is to act like a brake. Put the brakes on cell division. They are going to encode the kinds of proteins that keep a cell from going through a checkpoint. So these tend to be negative regulators of cell division. So they're either negative regulators. So what happens when I do that is my hand touches another part of the screen. Okay. So these tumor suppressor genes can be negative regulators of cell division, or they might be things that promote apoptosis. The normal function of a tumor suppressor gene is that it slows cells down from dividing, or it promotes their death. It either is going to prevent proliferation, or it's going to prevent cell survival, which is a different way of saying promote apoptosis. We call those group of genes tumor suppressors. And their name comes because their normal job is to prevent cancer from happening. Because these are breaks, you have to lose both copies of them to get cancer to be more likely. So, say normally we have we1 would be an example of this. We1 is a tumor suppressor gene, and we'll see a picture of that in a few minutes if you've forgotten. But it's supposed to put on an inactivating phosphate on a CDK, and normally there's going to be 2 copies of We1, and if a mutation messes up one copy, the other copy that's still normal can still do the job of negative regulation. So if we compare this to the waiter example, maybe you've had a job like this where somebody just doesn't come in that day, and then the other person has to work twice as hard, but they technically can cover for that. Technically, they can keep the brakes on cell division. But if you lose both copies of a negative regulator, now there's definitely not enough to get the job done. And if you've got 2 inactivating mutation, that removes the brake. That removes the brakes. And then the cell is going to divide too often, like we saw with We1 mutants, or the cell is not going to die when it should. It's not going to undergo apoptosis when it should. So a tumor suppressor gene mutation tends to be recessive. Those tend to be recessive mutations where you've got to have 2 loss of function mutations to have the problem become apparent. Those are tumor suppressor genes. So then the question becomes, how do you lose gene function? And this one's much easier to understand. How do you lose gene function? So any kind of mutation that is going to mess up production of the protein, that's a loss of function mutation. And some people are starting out with 1 good copy, 1 bad copy of a tumor suppressant gene. If they get gene sequencing and they find out that they've got a loss of function mutation, we tend to call that a risk factor. It doesn't mean that they have cancer, but they have some kind of mutation that's making them more prone to cancer. So they have a risk factor for cancer. So this situation is kind of halfway to losing a tumor suppression gene. They've still got one good copy that's taken up the slack for the bad copy. But anything that happens to the remaining normal copy of that gene to cause it to be lost can result in loss of tumor suppressor function. So you could have a good copy and have it silenced. Just because you have a good copy, if you can't use it, if that copy can't be expressed and used for transcription, you might as well not have it. You could have a second mutation in the other copy. Or let's not call it a second mutation. Let's call it a new mutation. You could have a new mutation in the good copy. So now we've lost that good copy also. We could have a deletion of the good copy. And this is still a deletion of the good copy. It's just it lost the whole chromosome it was on. These two really are the same thing. There's been a deletion of the good copy. So remember that to truly lose function of a tumor suppressor gene, it takes the loss of both copies. And this figure is assuming that a person is starting out with only one good copy. The ways that they lost this copy would be these same ways, but you have to lose both of them to lose the function of a tumor suppressor gene. So before we have our break, I think we'll have our break after this picture right here. So whether the mutations are in proto-oncogenes or in tumor suppressor genes, if the mutations are changing any of these pathways, that can lead to cancer. And the big takeaway of this is that any gene, any kind of gene involved with proliferation, detecting DNA damage, alterations in growth, or control of apoptosis, call that apoptotic control, the that do all of these things are really common to be mutated in cancer. And we can divide all this group of genes into either they're putting on the gas for cell proliferation and survival, or they're putting on the brakes. They're either going to be proto-oncogenes or they're going to be tumor suppressor genes. So right now I'm gonna kind of stop for a minute because I've been talking for a long time. And if you guys have questions so far, go ahead and ask them. Or if you need to go to the bathroom or whatever you need to do, I'm going to take a break for a few minutes. Okay, so, oops, I have my mic on. So I'm going to talk. So, what I want to step back now, and I've been talking about a lot of molecular detail stuff with proto-oncogenes and tumor suppressors, but I'm going to kind of zoom out and return to thinking about cancer in a general sense, and not just any cancer, but colon cancer. So, colon cancer is an inappropriate cell division of the lining of the colon, and we can see here that this is the lining of a person's colon, which may not be immediately recognizable, but this is kind of the normal appearance of colon epithelium. So it ought to be kind of smooth and shiny, and it ought to be lying down on a flat surface. But we can see here that this epithelium is kind of merging into a region that we call a polyp. And the polyp is an abnormal growth of colon lining. And so the way that we screen for colon cancer is people have colonoscopy, which is where they have a camera examination of the lining of the colon to look for polyps. And when you see a polyp, that's kind of a precancerous growth. And those can be removed. And if people tend to have polyps, they ought to have more frequent colonoscopies to monitor for that. So over here we can see this is the normal appearance of lining of the large intestine of the colon. That's the normal epithelium. But then up in here, the polyp is not having the normal structure or function of colon lining, and this would be precancerous. So the way that colon cancer, if we look at a network of proteins that are involved in colon cancer, it would include these kind of genes. So let's put a title on this. Stuff in the green boxes indicates genes that encode for proteins that have shown to be involved with colon cancer development. And I would say before you had this class, this picture would just look so awful that you would just skip past it. But this is interconnected signaling pathways. And if you look at what some of these genes are, we have talked about what they are. So here we've got RAS. So RAS protein that's involved in MAP3K pathways and is being signaled by receptor tyrosine kinases that detect growth factors. So detecting a growth factor through an RTK and activating RAS and activating MAP3K pathway, which is here, is able to change gene expression in a way that promotes cell proliferation. And that was a pathway that we kind of learned about. Or we could look at, oh, here's cyclin. We talked about cyclins and how they promote cell proliferation. We talked about P53. We talked a little bit about Wnt. So for people that study colon cancer in detail and want to provide treatments and prevention, everything on here relates to either promoting cell proliferation or promoting cell death in some way. And all of these types of genes, types of proteins, um, are involved in some way in formation of colon cancer. Some of them would be proto-oncogenes, some of them would be tumor suppressor genes. But together, they are changing what does an epithelial cell in the colon do. Is it going to divide more? Is it going to die more often? This would be your network of genes involved in colon cancer.
Can you go back?
Uh-huh.
Yeah, what are the red dots specifically?
Oh, good. Such a good question. So the red dots are proteins that they're not giving a name to. Let me find an example. Um, so if we look just at this pathway right here, there's a receptor tyrosine kinase that binds an extracellular growth factor, and that would be activating red dot, which is actually a RAS-GEF, the guanylyl exchange factor. And so what that red dot is it is an in-between protein that they're not bothering to name on here because they don't have enough room. So yeah, such a good question. So that particular one would be RAS-GEF. And then we know that RAF activates a MAP3K. That would be a MAP3K. That would be a MAP2K. This would be a MAPK. And then the MAPK has targets that change gene expression. So red dots are other proteins that actually are involved, but they're not naming them. So if we really wanted to know all the genes and proteins that are involved in colon cancer development, it would be the green boxes and the red dots. So one of my goals for this class is that you could look at one particular pathway and understand how that one leads to changes in cell proliferation. And you actually do know enough to do that if all of the proteins were listed here. The particular ones that we have talked about the most in this unit are the cyclin pathway and then P21 and P53 pathways. This kind of thing will not be on the final, but individual parts of the signaling pathways could be on the final. And I'll talk about those more in a few minutes. But one particular pathway that is very well studied and very much involved in colon cancer is the Wnt pathway. And so what we're looking at here, what we're looking at here is the detail of this particular part of the interconnected pathways. So Wnt, remember, is a particular protein that signals for cell proliferation in colon epithelium. It's an extracellular growth factor, basically. Or it's actually a mitogen. Wnt would be a mitogen that's an extracellular signal that leads to proliferation of gut stem cells and division of the colon epithelium. So we see here that when Wnt is present, Wnt is the red dot, it's binding to a GPCR, and it will activate the GPCR, which then activates G proteins that are not shown here. So there's a lot of stuff in this pathway that relates to G alpha, G beta gamma, and some of those in between steps that then are going to activate a particular signaling protein to block this complex that contains a protein called APC. And what APC had been doing is holding on to and sequestering, holding on to and causing degradation of beta catenin. And in this case, beta catenin is a transcription factor, transcriptional activator. And when this beta catenin is stable and not degraded by this APC complex protein, it's going to turn on genes that cause proliferation. So when Wnt signal is present, it's going to lead to transcription of genes that cause proliferation of colon epithelium. So one thing that I will that I kind of want to practice here is looking at what these different proteins do and classifying them as, are they a proto-oncogene or are they a tumor suppressor gene? So I think where I'm literally asking you to do that is at the end of this, toward the end of this slide set. So looking at all these different colored blobs, Wnt, the receptor, the signaling protein, the APC complex, beta catenin. For those, everything I'm putting a check mark. Take a couple of minutes and see if you could classify those as proto-oncogene or is it a tumor suppressor gene? So when you're looking at classifying proto-oncogenes versus tumor suppressor genes, it's really helpful to look at what's the function of the active protein. What's the function of the active protein that gene encodes? So ask yourself, if that protein is around and doing its normal intact function, is it pushing the gas or is it pushing the brake? And so in this case, when Wnt is around, it is promoting cell division. So Wnt itself is a proto-oncogene. Its normal basic function is to promote cell division. We would also say that the GPCR that is binding Wnt, when it's activated, its function is to promote cell division. So it would also be a proto-oncogene. This protein when it's active and signaling, it's blocking. See how it is blocking this protein, but that essentially is going to promote cell division. So this situation is a little bit harder to interpret. But when this protein is active, it is removing a brake on cell division. So this one also is a proto-oncogene. When it's active, it's working to promote cell division. So it's proto-oncogene. This one, we see it here in its inactive state. In its active state, it's working to block cell division. So when APC is in its active state, it is putting the brakes on cell division. So APC is a tumor suppressor. That one is tumor suppressor. And then we could look at beta catenin. When beta catenin is active and stable and actually doing its job, it is a promoter of cell division. So it too would be a proto-oncogene. And these are just like 5 examples, and there's other proteins not shown here. But what this means is if there was one gain of function mutation in Wnt, that would be enough to promote cancer or push a cell toward cancer. If there was one gain of function in one copy of beta catenin, that's enough to push a cell toward excessive cell division and toward cancer. But for something like APC complex here, that's a tumor suppressor, and you would need to lose both copies of your APC gene to promote cancer. So one of the things I'm going to want you to do on the final is to look at look at cell signaling pathway and figure out, is that protein a tumor suppressor or a proto-oncogene, and kind of classify proteins and the genes that encode them as being tumor suppressor genes or proto-oncogenes. So while we got 10 minutes, I want to make sure that I give you info about the final. So your final exam is going to be 150 points. It's a 150-point exam, so it counts more than regular exams, and it is cumulative. It's cumulative, but it's focused on cancer. It does not have sections for old concepts and sections for new concepts. It's just all in one test, and it's focused on cancer and cancer mechanisms. So the kinds of things that you need to study the most are CDK cyclin pathways. Study CDK and cyclin pathways and their regulation. That's going to be a big part of this exam. There are going to be questions about proto-oncogenes versus tumor suppressor genes for you to classify proteins in networks as they're doing one or the other. You also will want to look at P21 and P53 pathways. That's back in cell cycle regulation section. And those pathways and how they connect with cyclin CDKs, you'll want to look at that as well. Okay. And then the other major pathway that you will want to review in some detail is caspase activation that then leads to apoptosis. Those This is your like molecular detail kind of thing. Something else that you will have, well, and also Wnt pathway. There will be this same picture that we just looked at Wnt pathway, but that will be provided on the exam. So the picture is provided on the exam. This same picture is going to be printed on the exam, or inserted as a picture into Canvas, or however I have to do that. Also, let's see here. The details of these will also be provided. So the picture of cyclin CDK pathway activation and P21, P53 stuff, that picture will be provided on the exam. Caspase general details are not. That's something you need to kind of remember, the basic steps. Something else that I do on this particular exam is that you will have a new pathway that you haven't seen before. There's a figure of a new pathway that we haven't studied that you need to interpret. It's a cell signaling pathway, and it leads to cell proliferation, but it's got proteins that we have not particularly studied before. So, and generally students do well on that. I just want to tell you about it in advance so that you're not freaked out when you see it. It's a new pathway, and you interpret what's going on and make predictions about different alterations, what would happen to cell proliferation in that pathway. Okay. So when you're studying for this exam, you mostly need to study the new material, but the exam also includes information from the past sections of class about protein structure and function. So from Unit 1, protein structure, function, and regulation. It includes stuff about protein trafficking a little bit. Protein trafficking, and like how do proteins get to their destination. Protein trafficking and transport. That was from Unit 3. And also stuff about cell signaling. So cell signaling basics like G protein and RTK pathways. So you still need to know the basics of G protein pathways and RTK pathways. This exam does not have, well, the paper version of it does not have that much multiple choice. It's a lot of short answer, and it's a lot of looking at pathways of these various types and making predictions about what's going to happen if this mutation or that mutation, and looking at these specific pathways. It is going to be on Canvas, and you are going to need lockdown. I can't make this smaller. You are going to need lockdown browser to do that. So I've put on Canvas a link to a practice quiz that will let you practice using lockdown browser. This exam is going to be on December. What day is it? December. I can look this up. Hang on just a second. Whatever date is actually listed in the schedule. There it is. Okay, so it's Monday, December 7th at 1 p.m. It'll be Eastern time. So Monday, December 7th at 1 p.m., the exam will open up on Canvas. You do not want to be late for this. This is a I wouldn't say that this is a very difficult exam, but you want to have enough time to complete it. So it'll be open Monday, December 7th at 1 p.m. until 3 p.m. And you do have to do it during that time. It is not open note. It is not open book. And when you use the lockdown browser, you also have to have a webcam on that will be like kind of monitoring what your eyes do and where you go and if people come in behind you and things like that. So I don't love having to give the final this way, but that's the times that we're in. So you do want to make sure that you start it on time and so that you have enough time. Um, and it's not open note. Even if you had, I mean, I'm giving you the figures and different things. So even if you had notes, you wouldn't, it's not that kind of exam that you can just look stuff up. You need to know how the processes work. Um, what, what other questions do you all have about the final specifically?
Are there going to be more multiple choice than you usually have on it since it's online?
It usually, um, it usually only has 15 multiple choice in the paper format. And I don't know right now if it's going to have, I mean, I'm leaning toward not adding multiple choice because a lot of these are just short answer questions. Um, so it's what I'm thinking is it's going to have a lot of questions where I give you this figure and you look at the figure and then there's several short answer questions about the figure, or once in a while like a describe or explain like a couple sentences. So um, in the paper version of this final, it would be more writing and less multiple choice. And I'm going to try to just get it into Canvas in the same manner as it would have been on paper. So that also means that it won't be auto graded at all. These will take me a while to grade the way I'm planning on setting it up. Some other things to be aware of. I decided that your final papers are going to be due December 3rd, not December 1st, because I do I do myself want to kind of look through them and make some comments about things to watch out for or be aware of. And I haven't gotten. I'm like have a lot of stuff to grade over Thanksgiving, so your final papers are going to be due December 3rd. And when you're working on your final papers, make very sure that you are using all of the CSE format in your references and in your in-text citations. And I'm wanting to go through your drafts and kind of look at how you have it now and make some reminders there, but make sure that your final papers are good. So I know right now I have given you all basically completion grades of, oh yeah, you turned in the TBE list and you turned in your uh, article choices, and a lot of you have 100 on the paper project right now. Um, it's not going to be very common that that grade stays at 100 for the whole category. So your paper grades are going to decrease what your average is right now, just because I'm actually going to grade the paper for quality and not just completion like these other assignments. So I wanted to let you know that just because you have 100 on everything on the paper up until now, I don't grade the papers for completion. I do grade them for quality. So make sure you spend some time polishing up your papers and using the feedback that people gave to you. And then this exam, you need to study for it. You do need to study for it. Unfortunately, it's a kind of later exam on the 7th. If you guys have problems with the lockdown browser, I really want you to test that and not like wait until the morning of the test and like try to get all that figured out on that December 7th. Do that practice quiz, and then if everybody that does that in a timely manner, you'll get like 5 bonus on your final, so that you can make sure that you've got the browser working and the camera and all of that. If you have some kind of issue that you can't load the lockdown browser or it's not working, I need to know that like 2 days ahead. Not at the last minute, because I'm not good at technology issues on the fly. You all know that by now. But if you have any problems with the browser, or if you have any questions about anything over the break, just email me. I will be probably not checking email Wednesday through Friday, but after that I will be. So I've really enjoyed teaching you guys this semester, and those of you that are going to have micro in the spring, I'm excited to teach you again there. But you all have been a bright spot class for me this fall, and have cheered me up, and you've had good attitudes and generally good attendance. So I really appreciate it, and I hope that you have a great break and that you get some rest and that you also make time to work on your cell biology stuff. So if you have any questions, just stay on and I will stay on and see if you got questions or things that you need to talk to me about. But so much, and I will be checking email to see if you all need anything, and see a lot of you in the spring. Here we go.
Hi, Dr. Rom.
Hey, I'm trying to get my camera on. Hang on just a second.
Okay.
Tired of being a disembodied voice. Ah. Dang it. Actually, like, hang up and then rejoin Teams is going to be the easiest way.
Okay.
Sorry. Josie, I don't know if you can hear me, but I can't get out of Teams on the iPad. I don't know what the problem is. So
Yeah, I can hear you.
I don't know how to stop recording it or can't do anything. So frustrating. But I would like to not record this conversation if possible.
Yeah.
There we go.